Skin Cancer Dynamics, a presentation by Professor Gershom Zajcek, Faculty of Medicine, Hebrew University of Jerusalem. Recently I started publishing presentations on cancer dynamics, and they are published in ResearchGate. I already published 3 chapters. We are the first 2, and then here you have the 3rd chapter. And currently I am describing or discussing chapter 4 of cancer dynamic in the epidermis. So there are definitions which apply to all these units, and cell is an indivisible unit of life or atom of life. All cells come from cells, and the tissue proliferative unit, TPU, is the atom of histopathology. At its origin is a stem cell. Above the stem cell are the stem cell progeny. Stem cell division is associated with cell displacement toward unit periphery, and TPU is monoclonal. So here you have the tissue proliferative unit, and there are 3 major states: stem cell state, progenitor state, and end cell stage. And stem cell division is associated with differentiation of the entire unit, and as the cell move, they differentiate in this state. So the arrows here describe the differentiation of this unit. The arrow do not push, neither pull anything in the unit. The unit streams. There is no mechanical factor which is responsible for this streaming. And here you have the cell turnover. Division of the stem cell is the birth of the unit, and these are the progeny of the stem cell, and as they move or stream, they differentiate until they reach the end cell, and the end cell actually is the last state, and it will sooner or later die. So we are talking about epidermis, and this tissue proliferative unit is applied here to the epidermis, and here you have the epidermis, all the state of the epidermis, and at the bottom we have the stem cell, and as the stem cell divides, the entire unit streams upward. Now, what is important to realize that this unit maintains homeostasis. So, in order to maintain homeostasis, the end cell creates chelons which inform the stem cell about its state. So, chelons are polypeptides produced by a tissue that inhibit mitosis in the cells of this tissue. So this end cell inhibits mitosis of the stem cell so that the entire unit maintains homeostasis. So in order to, it was already published last century, and it's a very important concept to understand the dynamics of cancer. So this definition applies also to cancer, and here you have how cancer evolves. So it starts with a founder mutation, and they hit obviously only the stem cell, and once the stem cell mutates, its progeny mutates, also receive this mutation because they are the progeny of the stem cell. Now, in order to show you this, how this Kalon functions, we do this hypothetical or experiment. We kill the end cell. Next state, the end cell dies, and this information is now provided to the stem cell. The stem cell knows that the end cell died, and therefore its progeny starts dividing faster, and we have two green cells which respond to this irradiation. And once this end cell is restored, it again is inhibiting the proliferation in the stem cell, and we get the unit back again in the same time. It's a very important concept, and here you have the experiment which shows how this Kalon functions. And another experiment which I try to show you is what happens if we irradiate the unit and kill only the end cell. So what happens, here you have a normal unit, and we start irradiating this end cell. Obviously they die, and the Kalon inhibitor inhibits less the stem cell, and the stem cell responds by producing, here in this example, two progeny, and we are talking about a chronic irradiation, which means we irradiate all the end cells so that all end cells are dead, and therefore the Kalon inhibition does not work on the stem cell, and we get this response. So, conclusion, this experiment shows that irradiation may promote tumor growth, because these greens are actually tumor, and their abundance is now increased because we irradiated the end cell. I hope you like this idea because it is applicable to all cancer. Enjoy.